I think there was a question from behind.
Thanks. Trevor Howe from Janssen. I just want to agree with my bearded colleague over here that the model that you proposed of unblocking the path of drug discovery, that's a very reductionist linear view of drug discovery, familiar to me 20 years ago when I joined the industry, and I worry about unblocking an old problem. There's a sewage system in London built by Bazalgette during the 19th century which solved the problems of Victorian sewers and for the foreseeable future of 1 million or 2 million people. London now has a population about the size of Belgium, and unblocking those sewers isn't actually going to solve the problem. So I would strongly support the idea of examining phenotypic screens and making sure that there is a rationale for translation of those. Otherwise, we're just going to screen the same things we've always screened with a different compound collection, but
don't get me wrong. Nobody said it's going to be excluded.
Okay, good. So that's one thing I'd certainly encourage. The other thing I'd certainly like to see, and I think that's in work package 2. But the other thing I'd certainly like to encourage is not a view of relatively conservative chemistry, because we've kind of done that for the last 20 years. And I think it's, in order to make a difference, we really need to break out of the so-called rule of 5, which isn't. It's an observation, and we really need to be adventurous about the chemistry and not constrained by what we've always done, because it's not working.
Agreed.
Then I look forward to this proposal spending 80 million euros extremely wisely.
Okay, so there were several questions on this side.
So Jörg, Nyström, AstraZeneca. So I have a question regarding the building up of the collection, quite specific one. So how much of the budget do you expect, or have you decided, will go into these 200,000 compounds? That's one question on that. So will that public collection be open to the public, the structure information, everything?
That's the question. Budget-wise, the original idea is to split the budget that we have in 2 parts, really by half. One part for the screening center, the other half for library generation, so that you have a number for this activity. The second question slipped my mind, actually.
So will the structures in the public library be open?
Will it be open? Since the entire exercise is again about generating value, maybe about providing a model for sustainability for the screening center. Yeah? We have an idea, at least, and it's only an idea, it also needs to be clarified in a full project proposal. We have an idea to keep that as closed as possible. Mm-hmm. Yeah? The project proposer has an interest in publishing the realization of innovative chemical concepts, synthesis concepts, and that should be also the goal of this initiative, which does not mean you have to disclose all the structures. Yeah? That provides also the screening center after the 5-year funding period, which at least one asset they can build on a model for sustainability, and that's a unique compound library that has been synthesized in the context of this project, as was one proposal.
I see an issue here, at least one.
There are probably plenty of issues coming up.
If you think about it, you have 4 or 5 suppliers to the new library, and they don't know what they're proposing to build the library with. They may overlap in their approaches, they may overlap in structure. So how will that be avoided in order to create the best value?
Well, every single unit being involved in the realization has to know, of course, what the others are doing, and making sure that one proposal is realized only once. That's pretty clear. Was that the
Yeah, but I guess it's 2 stages. One is the grant stage of the application, where you present your idea. This is the way we would like to build the new library, these structure types or synthesis or reaction types or whatever. And you get approval on that, hopefully. And then you're in the consortia. but some it has to be coordinated. You may need to amend these plans, or I don't understand how that will function, both from a grant perspective and then post-grant work.
Yeah, that's going to be a deliverable of the applying consortium here. Yeah? We have to see what the processes envisioned by the consortium are. Yeah? They have to solve this technical issue. See what I mean?
I hear what you say, yeah.
Okay, okay. It's close enough.
So it's further along.
Hi, Jordi Quintana from Barcelona Science Park. I have two questions related to the consortium, how to build it up, and if you may provide some clues on that. The first one is that you are proposing two subtopics. The question is that if there will be a preference for those two subtopics being linked somehow, since one is referring to chemistry, the other one to screening, and at least from the chemical biology community, we would think that the two should be interlinked. Okay, that's one question. The other one is more on the screening center as it's proposed is one single site. And if we want to aim for innovative assays and innovative chemistry, that may mean that that screening center will need to be developing those screening assays in the screening center, and therefore we are not going to bring up the knowledge and the innovation in assay by the biology community in Europe. So my question formally would be if a network of target centers will not be considered, and therefore the application or the applicant consortium should aim for one single screening center.
I think it's pretty straightforward in the text, in the document, as we have agreed upon. And there we are talking about a single screening center. It might be two sites. I think that is the way we have phrased it in the text. And you can think of it also in a different way. You can spend a lot of your budget managing a very complicated network in that aspect. There's certainly also benefits, no doubt, but it has the disadvantage of that you have to spend a lot of time and effort bringing the bits and pieces together. And we lose a lot of the operational efficiency that I was talking about, really to come up with a meaningful number of projects being performed in a very limited period of time and within a very limited budget frame. You have to balance all these things. On top of that, fragmenting these activities also means having a more complicated way of information management behind confidential information, which is also an issue. It complicates things. That's why the entire EFPIA group agreed upon looking at this balance that we prefer clearly sticking with a centralized unit for screening. I agree. In the ideal world, it would be maybe a different scenario, but we have to make compromises here. We have to find a balanced way to approach that. And we still have possibilities to implement also innovative assay technologies by bringing in partners on a project-by-project base. Yeah? There are lots of possibilities within that framework that I've shown, having this core technology platform, but also having the possibilities of having public partners on the outside. So I see this is a good solution with a balanced way to approach this problem.
And with respect to the chemistry, the two subtopics being merged, or?
I think for looking at the complexity, it makes sense to separate the two in terms of the application process. If you apply and you can refer and put references into another application in terms of the library generation and the screening center, that would be very useful, and it might be a unique selling point for that consortium, but it's not necessary for the application process per se.